distribution map is surface data and model with the Maxent model. But when we plotted data from GBIF, so occurrence data from different sources, we could see that lots of data were outside such distribution ranges of the habitat. So what we aim to do with our use case was to complement the system using not only vegetation service data, but also data coming from museum collections and citizen science from GBIF. And we modeled them through machine learning algorithms, and we produced this distribution map of one particular habitat, S22, which was our use case study, and which is a habitat present in the mountain region and dominated by ericoids and other woody species. And so what we did in our workflow was to choose this habitat and then taking the diagnostic taxa of these indicated in the classification, and then retrieving data from GBIF using the R package RGBIF, and at the same time also retrieving environmental data from Kubernetes or WordClean, and then processing and cleaning all these data and putting them together in a data cube. And also preparing pseudo-absences points based on the present data from GBIF and using this random method with a radius of 100 meters. And then we used a supervised ensemble model built on JM, DLM, and random forest models evaluated with CSS forest. So we then had prediction maps of all these 8 diagnostic species. And then so we aggregated them to have derived habitat areas for our use case habitat. And then we also validated it with the EVA vegetation plots. And in the end, we compared our results with the AONIS habitat probability map. And what we could see is that ecologically speaking, the presence of this habitat was in agreement with what the AONIS system was saying. It's just that when we were plotting at 1 kilometer resolution, our results, we could see that it was overlapping only mainly in the Alps, as you can see in the 1 meter resolution. While in the other regions of Europe, it was not really, we couldn't see it. And when we were lowering the diagnostic species to 3, actually we could see that it was also coming from in other regions. So we could see that the overlapping is mainly in the Alps. And then what we could get from this project mainly was that plotting all this data at the bigger level of the AONIS level 1, using all the diagnostic species considered, so 348 is just too much for the processing. And then so we chose the level 3 of the AONIS classification, which had way less occurrence data. And also what we could learn from this is that the museum collection data are often incomplete in terms of coordinates and uncertainty in coordinates. And also we have way less observations in comparison to surveys data and citizen science, and also often clustered. So not the best data for the distribution map, but still taxonomically correct and still useful. And so also useful, for example, the pipeline processing at the botany department, because with now this workflow, we can decide where to allocate the resource allocation for the collections, or also how to support monographic studies in the Annona group, and also characterize a tropical forest, for example, the upper Amazon. And thank you.
We have 1 minute of questions.
Many questions. Good, then we have more time later.
You can stay for a minute. Is there anything you want to go on?
Next one.
Okay, then we can prepare the next speaker. So we have time. Next, bioprepr.
Anything?
Which
Okay.
sorry, what button? Ah, perfect. Thank you. Yeah. Hi everyone, my name's Charlotte. I'm a third-year PhD student at the University of Essex, and today I'm going to be speaking to you about something I've been developing called ukbioprepr and our package to support reproducible preparation of environmental data for biodiversity modelling in the UK. So my PhD project involves a lot of UK-wide biodiversity modelling, so I'm really interested in predicting how nature is going to respond to things like climate change and land use change. And I probably don't need to tell a room full of you guys, but there's a lot of steps to biodiversity modelling, one of which is all to do with data. In the UK, we're really lucky to have a vast range of open source accessible data sets on biodiversity, including the National Plant Monitoring Scheme and the Breeding Bird Survey, who operate using citizen scientists who are allocated a 1 kilometre grid reference square for repeated surveys throughout the year. And the same is said for environmental data. We've got a really wide range of open source accessible high resolution data products available, covering land cover, climate variables, and soil properties. However, when I came to use these for my own work, no. Oh my goodness. Okay, when I came to use these for my own work, I realized that these were really fragmented and inconsistently formatted, both spatially and temporally. I also realized that there was lots of differences in coordinate reference systems across these products. So I found that some of them were in British National Grid. So you can see here how that lays out across Great Britain and Northern Ireland. And I found that some had data products for Great Britain in British National Grid, and then Northern Ireland in Irish Grid on its own. The biodiversity data sets that I spoke about and wanted to use, I found that sometimes they had their grid reference locations in British National Grid, some in Irish Grid, and then some actually had the same locations in both. So I was left trying to figure out what I was meant to do. Because when you try to extract data using different coordinate reference systems, it can cause inaccuracies or uncertainties. So I started looking for existing tools. This fantastic paper that was published 2 years ago published a list of 60 R packages, all to do with ecological and biodiversity modelling. But of the 60, only 3 of them were to do with environmental data preparation. And I found that they all had the same issues that I was facing. So ultimately, they weren't appropriate for... Oh, this went so far. Ultimately, I found that those packages weren't appropriate for biodiversity data in the UK. And that led me to the creation of my package, ukbioprepr. I started off by trying to create ready-to-use data products. So these are the state of the original data products. And in the interest of time, I'm not going to go through them all, but you can see the differences in the coordinate reference systems and then the differences of available years. All the original data products went through substantial data engineering of cropping, aligning, masking, and projecting. all to create data products covering the whole of the United Kingdom in British National Grid, and then Northern Ireland just on its own in Irish Grid. It left me with climate data products on climate variables from the year 2000 to 2023, land cover for 2000 to 2023, and 2015 to 2023, and then soil properties at a range of depths. So you're probably wondering about why does land cover have two sets? This is because the original data products only existed for certain years. And on top of that, through those years, the land cover classes, they changed due to new methodologies, different data. From 2015 onwards, the land cover classes were consistent. So I used that as a basis to create a modern set of land cover products with more cover classes. And then I was able to aggregate some of the other classes using the technical details that were provided by the original creators. So you can see, for an example, the montane habitats were aggregated with some of the other like associated cover classes to create an upland class to give consistency from 2000 to 2023. And as well as that, to combat the temporal inconsistencies in the land cover data, I used a linear interpolation method to generate data for the missing years. So these maps show the percentage of urban cover for the years 2007, 2011, and 2015, with a bit of a zoom in on kind of the area of Greater London. 2011 was the year that was created using the linear interpolation method, and from the graph you can kind of see how that happened. So yeah, once I had all that and had all these data products ready to go, they're uploaded in an online repository accessible through functions in the package. First kind of functions are fetch functions, and these return ready to use raster files for selected variables based on the location and a time frame. An example of this is the fetch climate raster function, and this has an additional customization for users where they can choose a time aggregation of seasonal, monthly, or annual for 4 different climate variables covering temperature and precipitation. The other type of functions are extract functions. These return values on different environmental covariates using either X, Y point data in a specified coordinate reference system or using those grid references. These functions, the grid references are automatically detected as either in British National Grid or Irish Grid. So it means it's a really seamless, it has a really seamless and easy integration with all those existing data sets that I mentioned earlier. So to demonstrate the functionality of ukbioprepr, I carried out a case study estimating the occupancy of hyacinthioides non-scripta, which are common bluebells. These native wildflowers might be susceptible to climate change. And so I investigated this by mapping the baseline occupancy using data from the National Plant Monitoring Scheme, and then predicted how this occupancy probability would change and how the suitable habitat area would change under different climate scenarios using relative concentration pathways, which are effectively a best case, middle of the road, and like most pessimistic case of how the climate will change in the 21st century. Using the extract all function from the package, I incorporated these environmental covariates into the model, covering soil properties, land cover, and climate variables. And then this is the baseline estimation of occupancy probability. So there are white areas in this, and this is because there's no probability predictions. The original soil data products, they don't have coverage in urban areas, and there are ways of inferring it through like machine learning or interpolation methods, but I decided to leave it up to the user to choose what they wanted to do with that. And then this is the occupancy probability change under different temperature scenarios across the top, and then precipitation scenarios down the right. You can see the most optimistic scenario in the green box. There's not really that much of a change. In some areas it actually increases, and this is contrasted with the most pessimistic in the red box, where some areas decrease by 8%. And then this is the weighted suitable habitat areas under the different climatic scenarios. Under the most pessimistic, there was a decrease of almost 12.5% away from that estimated baseline. So just some take-home messages. Sourcing and preparing environmental data is a really important step for biodiversity modeling, but it's really, really time-consuming and can be technically challenging. ukbioprepr provides streamlined functions to access and use environmental data products tailored to UK biodiversity data and has applications such as scenario-based modeling. And this framework of having pre-prepared, ready-to-use high-resolution environmental data products is a framework that should be considered by other nations in order to expedite biodiversity research and action. So thank you so much for listening and for dealing with the technical difficulties. If you're interested in the package or the paper, do reach out to me, drop me an email, connect with me on LinkedIn, have a chat with me today. At the minute, the paper for this is under peer review, so the package isn't live. But if you do want to use it or have a look at it, just have a chat with me and I can sort you out with it.
University of Gießen in Germany with a talk on integrating GBI and accessible and powerful R package.
Please try to hang out with the laptop. No, on the screen. It's very sensitive with this. Okay.
So for those of you who are interested in calculating biodiversity indicators, but you don't like coding very much, this is to make your life a bit easier. So we all know we're in the middle of a biodiversity crisis, and we need a lot of biodiversity data. It needs to be reliable, it needs to be quickly available, and it needs to be analyzed in a repeatable manner. And this is not always the case. So the BCube project was created partly to solve this problem by creating standardized and automated workflows. And the core of this project is the species occurrence cube, which was introduced earlier by my colleague Lisa. And the basic idea is that it's species occurrence data presented in a standardized and aggregated manner. And BCubed has a whole suite of tools available. We've already heard about pdindicatoR. And now I'm going to talk to you about B3GBI. Now this package is all about user friendliness. So B3GBI stands for BCubed General Biodiversity Indicators. So it's all about common metrics that you can calculate using only occurrence data. And it has a very simple three-step workflow. So first of all, you import your cube. You can get your cube from the gbif.org website, and we've already seen how to do that. That's probably the easiest way because it's a graphical user interface. You can also get one using the GBIF package, or if you're interested in using simulated data, you can use the gcube package, which is another output of bcubed. Next, you calculate your indicator. Again, this is just one line of code. There are 16 different metrics available in the package at the moment, including richness, evenness, and a number of others. There are uncertainty calculations integrated for time series. It's not yet integrated for maps, but this will be coming eventually. And you can also use your own shape files to get a more specific shape. If you want to find out what indicators are available in the package, you just type available_indicators and you get a nicely formatted list, which includes usage information. And you will get a specific output, which includes metadata, and it will print it out nicely for you when you type the name of your object. And the third step is to visualize, and you just use the inbuilt plotting command of R, and it will automatically send your data to the correct plotting function from the package. And you can very easily generate publication-ready output. So it's integrated with the ggplot package, So for those of you who are experts in ggplot, you can add your own custom code on top of it. If you're not, there's still plenty of options available within the package to change your visualization. It uses our natural earth for your map data, so you get very nice looking maps. As I said, shapefiles are integrated, so you can get a more specific output if you have a shapefile of a protected area, for example. And I've just added a new feature, which is map view integration, so you can get an interactive map where you can zoom in and out, look around your map, or change your base layers. And this uses the plot_MV. So this is an example of an actual workflow using a cube that's included as an example within the package. You just give it a name, you run process cube. In this case, we're printing in observed richness map, and then plot your results. And it comes out looking like this. So if we want to refine this and make it a little nicer, we've already imported it. So we just add a few options to our indicator. It's a cube of Denmark. So we say, OK, we just focus on Denmark. We're not interested in the ocean occurrences. We want to use the finest map data. And we only want 1980 to 2020. And then we give it a custom title, a legend title. And we have a much nicer, ready to publish map. So it is also quite a powerful package. It supports 3 different grid reference systems. You can use any coordinate reference system that you like, as long as it's compatible with your data. You can add shapefiles. You can add any map layer that's available in our natural earth. There are lots of customization options. And of course, you can add your own ggplot code if you want to. There is documentation available for all the user-facing functions. There's a tutorial to get you started. And it has a modular design. So now we're not moving anymore. Oh, there we go. So if more indicators need to be added, all that's needed is the codes created for the indicator itself. And you just plug it into the package, register, and it's ready to go. So you don't need to change any of the core code. And finally, there is a graphical user interface available, created using Shiny. There is a web version online, which you can actually use without downloading anything. It has most of the functionality of the package. And you can also explore your cube within it. You can export your data. You can create plots and export them, things like that. So it's very easy to use. And that's it. Thank you.
Are there any questions? Are there any ways of putting detections or data gaps or make it more? Or is it somehow considered when calculating these indicators so they're comparable?
So, I mean, all the data cleaning and figuring out what data you want in your cube and everything, that's all done before you put it into the package. So whatever you have there in your cube will be used in the indicator. I mean, you can filter your data, you can take out the species you don't want, things like that. But the majority of that processing happens at the GBIF stage. So yes, there's the RGBIF package, which, as far as I understand, can now make cubes. But the easiest way is to use the website itself, where it has a graphical user interface. You choose everything you want in your cube, and then it builds it for you. And then you get a permanent DOI for your cube.
You can add what is environmental, but related.
So I don't have any plans for that at the moment, because the idea was to keep it simple so that it only uses species occurrence data. Maybe in the future. Let's see, it's still in development. So.
particular with the most responsible to prove it.
Thank you very much. Sorry for the long title. First of all, I need to thank the colleagues from UK Antarctic Survey who are at the origin of the work I will showcase today. In fact, I find the different presentation before really interesting and related. And here I want to start to speak about Galaxy-Ecology, which is something we are promoting since 8 years, with in fact a manner to package R script, Python script, Julia, or something else, or Bash, into a platform with open source and accessible to the web, and who allows you to have a particularly important reproducibility, and moreover, who allows you to compose your own workflows. For me, workflows is computational workflows, because I was from bioinformatics in a previous life. So I will just illustrate a little bit of that today on this work with Antarctic Conservation Showcase. So working on that, we were starting from R script and R package and some Python codes coming from colleagues working in UK, who develop, they are developing a PDF. They are creating a PDF file with a report on the ecosystem, state of ecosystem of Antarctica in their region. And we were proposing to test the use of Galaxy-Ecology to take all these scripts and try to make something with reproducible and accessible through the web. So we can have computational workflows. We can take the data from Copernicus or GBIF or something else automatically after selection by a user of some parameters and something like that. So the user don't have to know how to use R or something else, just the web form and things like that. So there is two main things we were working on this work. The main thing is how we can automate the production of such a report at the end of the workflow execution. The second one is, in fact, something we find with, in fact, a new way to contribute code to Galaxy-Ecology. So just to show you, I don't know if you know Galaxy. Galaxy is a web platform used since 20 years by colleagues from bioinformatics. So if you see previous slides, for example, speaking about issue regarding scale up, regarding the need of more computing powers and things like that, Galaxy is made for that because it's directly connected to high performance computing. So if you need to have a GPU cluster or a CPU cluster or big amount of memory, Galaxy can allow you to have this interface where you can execute your code in such infrastructure. It's one of the interests. The other is the web interface. It's accessible through the web and people can share data, history, tools, workflows to some colleagues or to the world. Just to illustrate, here we see a workflow for Galaxy. So each brick is a script, in fact. And behind each brick, we have a container. So it allows you to have real reproducibility in terms of entry and output of each script. And we manage all the dependencies in a manner you have big reproducibility. So yeah, I just want to showcase this because until this year, it was quite complicated to say Galaxy can be a platform usable by everyone, notably to execute workflow because it's a little bit raw. There is plenty of tools. It's quite complicated. And now we have a manner to create something really interesting. So here you see the form that people have to fill to execute Galaxy workflows, where we see that the user can select or not some steps. So he wants to have a figure or not. And he have example of what will generate the script, each step of the workflows. And he can choose or provide its own data sets or use existing data sets and specify some parameters. So he can facilitate, I don't know, the taxonomics, the geographic of the temporal delimitation of the workflow. Here you see the execution of a workflows. where you see in Galaxy the execution, which step are executing. You can access all the metrics for each step. You can share this to other people if you want. And at your right, you see an historical of each analytical steps you can reproduce if you want, and you can share with others. The reproducibility is really the core goal of Galaxy. So if you have questions about that, we can discuss. And now if I go to a technique, it's a horrible slide, sorry. Another thing is really interesting, and I think I can, for example, test for the R package we just see before. We can, in one day, create a component into Galaxy from a raw script, R, Python, or something like that, and push it into a workflow form so other user can use. This is really new because before, to create Galaxy tools, we need to have something like, depending to the scripts and the state of the dependencies, 2 days, 1 week, 1 month, 2 months, because we follow good practice to package all the dependencies using Conda Package Manager and automatic creation of containers, so we can reuse it behind Galaxy or just directly from other platform who allow user workflow. Now we can directly have R script, for example, or Python script, and use something with Jupyter Lab tools into Galaxy. So you deploy your Jupyter Lab, you test interactively, your code is working with input parameters and input dataset and generate the good output. Once it's okay, you just stop it. And then, after that, you can relaunch it, but in batch mode. So you just provide the data, the parameters, the Jupyter notebooks with your R code is executing, and you have your result at the end, and that's all. You don't have to code or something else, and you can share it to others really easily. It's a new way to share code. It's less fair because we have less management of dependencies and things like that. It's less reproducible, but it's not so bad. Yeah, okay, that should see you that in fact, after that, you have executed your Jupyter notebooks with the R code. You create a workflow, so you can do that in 1 day, and after that, you can share your preferred R package or R script with other people so they can reuse. Just to briefly mention the paper we published this year. After 8 years, we take our time to be sure that we have enough information about the interest of Galaxy for ecology. We published this amazing paper because the first author is Colin Royot, who was in thesis with me, and really made a wonderful work to explain not what is Galaxy or what is the tool, but a manner to think about sharing complete workflows, computational workflows in ecology, looking at good practice in ecological data analysis, and notably through something related to what we call an atomization generalization framework to automatize and to create units of pieces of code we can reuse into workflows and reuse and share through also open source license. You can test it now, but if you click on that, you will have access to the workflow. You can execute the workflow and reuse it and look at it. And just want to thank all the young colleagues who are working with me since 8 years to test things. And if you want to test things or go further, please go to Ecological Galaxy instance, the European one, and contact me if you have any question or something else. There is a lot of tutorials, a lot of things, and we are now ready to really test it intensively in any case in ecological data. Thank you.
Thank you very much. Now have like to ask all sentence you prefer the strict vegan variant. I also have some here. And as an incentive for all of you to ask questions, everybody who asks a question also gets some. So, this is,
Sí. Ah, no, no. Entonces, que si las podía leer.
So, oder Kochen has very fun us. You will of course get some stuff.
Thank you.
Thank you.
And yeah, you
Also,
already
there
have-
is two questions for Sean from the Zoom session. I don't know if you want to answer. The first question is, there is accessibilities.
So not for my package, but if you want to download a cube, you can use an API at the gbif.org website, as far as I know.
And the second one is, do you correct for the observation pressure and taxonomic bias, the change of the state into the observation euphoric indicator?
I don't fully understand the question, to be honest.
Really, I don't know.
I'm sorry, I don't understand that one.
Okay.
Are there any other questions from the room?
Also flexible to include another type of